Hi everyone, today we'll be going over Candidiasis. The most common cause of this infection is attributed to Candida albicans. This fungus is often characterized by its unique ability to switch morphologies between yeast and hyphal states. Candida albicans is normally an opportunistic pathogen. It's part of our normal flora. Globally, Candida albicans is the most common species to cause infection, systemic in particular, and most of the time, Candida infections typically fall under two categories, being localized or systemic. We will focus on systemic first. Candidemia is a very common healthcare-associated bloodstream infection. The CDC found a total of 1,226 Candidemia cases that were identified in 2017. They estimated that there were a total of 23,000 cases in the States. For the prognosis, this disease typically afflicts immunocompromised and immunosuppressed individuals, causing a 40% mortality rate. Patients who are afflicted with candidemia typically present symptoms similar to septic shock. This includes fevers and chills that do not improve with antibiotics, low blood pressure, fast heart rate, and rapid breathing. These symptoms can progress to kidney failure and shock when systemic infections go untreated. On the other side of the spectrum, we have candidiasis, which refers to a localized candida infection. This can be in the mouth, otherwise known as oral thrush, the genitals, the more common form being in women, also known as vulvovaginal candidiasis or yeast infections, or skin infections. The CDC estimates for localized vulvovaginal candidiasis, 75% of women will have at least one episode, and 10 to 20 will have complicated or recurrent vulvovaginal candidiasis. Symptoms for localized infections in the genitals include a burning feeling while having sex or while urinating, an itchy or painful feeling on or around the genitals, redness, irritation, or swelling around the vagina, and an abnormal vaginal discharge that can be either watery or thick and white. Symptoms for oral thrush include white spots in your mouth that have the appearance of cottage cheese, bleeding, cotton mouth, difficulty with eating or swallowing because of pain, and even a loss of taste. Most Candida infections can typically be fought off by our immune system, unless complications like immunosuppression occur. Persistent infections of Candida are usually treated with antifungal medications, the most common oral form being echinocandins, like fluconazole, and more topical forms like Miselex for localized infections.
Candida albicans is naturally part of our normal flora, found in our digestive tract, genitourinary tract, and is part of the skin, conjunctival, and oral flora. Statistically, Candida albicans is present in the oral cavity of about 75% of the population. Most localized infections will take place on mucosal surfaces. In systemic cases, there will be large amounts of Candida buildup found on filter organs. So how does this infection start? Most systemic Candida infections are of the host's own population of Candida albicans, but some infections are hospital-borne. Exposure can arise from the usage of catheters, intravenous injections, and even lung infections. The two major modes of infection include induced endocytosis, which is a response to the host's immune system, and hyphal penetration, which is invasion of epithelial cells. The morphology of Candida albicans helps with the persistence of infection. The hyphal form helps Candida colonize surfaces. The yeast form helps with the infection spreading or going systemic, where individual cells bud off from pre-established hyphae, kind of like how a cancer metastasizes. Hyphal form is able to pierce phagocytes, leading to intracellular infections. Morphogenesis, or the transition between morphological states, is regulated by quorum sensing as well as other extracellular factors. So now we come to the immune response. Like most infections, the innate immune system encounters the Candida first. In short, our first line of defense. In certain cases of exposures, our epithelial and endothelial cells have to deal with the pathogen first, often releasing DAMPs. Now, the first group of cells that arrive to the site of infection typically consists of macrophages and monocytes, dendritic cells, PMNs, or polymorphonucleated neutrophils, and natural killer cells. Professional APCs, like the monocytes, macrophages, and dendritic cells, are able to recognize Candida albicans and thus activate neutrophils and later immune responses using certain TLRs and CLRs. These receptors recognize fungal components like manoproteins, beta-1 glucans, as well as layers of chitin. These are pinged by these immune cells, and several pro-inflammatory cytokines facilitate the migration of neutrophils to the site of infection. We will go into more depth about these receptors and pathways later. Neutrophils are able to deal with Candida albicans in a number of ways. The fungus triggers the net process of neutrophils, which mediates phagocytosis of the pathogen. These immune cells also release oxygen and nitrogen intermediates, as well as hydrolases, limit nutrient availability, and express antimicrobial peptides. NK, or natural killer cells, exert antifungal activity through the secretion of granules like perforin. Perforin is an enzyme that causes the formation of pores on the membrane of pathogens. Also, NK cells can indirectly cause a feedback loop through the generation of certain cytokines that help recruit other specialized immune cells like the professional APCs and neutrophils.
Next, we'll go over what receptors are activated as well as which signals are sent to the rest of the immune system and how. The first category of receptors that we'll go over today are called toll-like receptors, or TLRs. The two primary TLRs involved in the clearance of Candida albicans infections are TLR2 and TLR4. TLR2 recognizes surface proteins, and TLR4 recognizes O-linked sugars. Activation of these receptors through the binding of manoproteins, such as those found in the filamentous state, contribute to the induction of pro-inflammatory signals via the MyD88, the MAL-mediated, and NF-kappa B pathways. The next category of receptors is the most well-known in the immune responses for the clearance of fungal infections, this family being C-type lectin receptors, or CLRs for short. The most important CLR during the innate response is Dectin-1. Dectin-2 and Minkle are other receptors that are important for the clearance of the pathogen, but these are more important after the adaptive response sets in. Dectin-1 is a receptor that recognizes beta-glucans found in the fungal cell wall and initiates a series of cellular responses via the Seq, CARD9, and Seq-NFAT signaling pathways. When Dectin-1 is activated, Seq, which is a tyrosine kinase, activates NFAT to produce IL-2, IL-10, IL-12, and COX-2. Seq also activates CARD9, an adaptive protein that downstream activates nuclear factor kappa B, or NF-kappa B, to produce IL-6 and TNF-alpha. If the inflammasome is also activated, NLRP3, which transcribes for cryopyrin, is activated to start producing IL-1 beta. The last category of receptors that respond to fungal infections are nod-like receptors, or NLRs. NLRP1 is a subset of NLRs that are cytosolic, able to assemble and oligomerize into a common structure known as the inflammasome. This collection of proteins activate the caspase-1 cascade, thereby leading to the production of pro-inflammatory cytokines, especially IL-1 beta and IL-18. Through the activation of the innate immune response, subsequently T helper polarization through cytokine activation takes place. Antigen-presenting cells like dendritic cells produce IL-12 and IL-23. These help to differentiate T helper 1 and T helper 17 cell types respectively. T helper 1 cells help promote cell-mediated immune responses toward intracellular pathogens. Due to Candida albicans' ability to penetrate endothelial cells and persist in some phagocytes, this is extremely valuable. T helper 17 cells help to protect against extracellular pathogens and mucosal surfaces. Again, as large amounts of Candida albicans infections take place at mucosa, like the oral tract and vulva or vagina, this is also extremely valuable. An interesting tidbit regarding T helper 17 cells is that depending on the location of the fungal infection, different cytokines are more prevalent. IL-17 is more commonly associated as a response to oral infections, and IL-22 is secreted in response to vulvovaginal infections. Now, how do these immune responses affect the infection? In most cases, the immune responses can clear the infection. However, Candida albicans can subvert our adaptive response formation. The yeast and hyphal forms of Candida albicans can skew T helper polarization. This subversion of T helper subset differentiation can enable fungal persistence within the host. Dendritic cells, which phagocytose C. albicans yeast cells, promote the development of T helper 1 responses, which leads to fungal clearance, whereas dendritic cells that internalize hyphae generate T helper 2 responses. This leads to fungal persistence. This trend was echoed when Dr. Bakke and his fellow colleagues incubated dendritic cells with yeast and hyphal RNA. The reason why T helper 2 responses lead to fungal persistence is due to the fact that T helper 2 is specialized in response of extracellular parasites, and the Candida albicans species spends some of its life intracellularly.
In conclusion, yes, a healthy individual can clear Candida albicans infection on their own. However, immunosuppressed or immunocompromised patients can struggle to recover. This is why we use antifungal medications like fluconazole and Miselex to help prevent infections. While several studies have explored how functionally distinct strains as well as morphologies affect the interaction with the host, the molecular basis for the observed phenotypic differences remains unclear. The field is awaiting identification of specific genetic and genomic variations that translate into relevant phenotypic changes in Candida albicans to affect the balance between commensalism and pathogenicity in different host niches. Difficulty in identifying critical components of the biofilm matrix that are responsible for antifungal resistance make it harder to treat these biofilms, making these infections especially dangerous. This high tolerance may be contributed to persister cells, which are highly tolerable to antifungals. But studying persisters is limited due to the absence of effective detection techniques. These gaps in identification, as well as lack of standardized testing protocols between laboratories, makes it difficult to gather and compare results. Flow cytometry may be used to help bridge some of these gaps. Green fluorescent protein stains labeling Candida persister cells that are still viable would help narrow down the cell pool from those that die in a biofilm matrix upon exposure to an antifungal. Ultimately, Candida albicans is a major problem in immunocompromised or suppressed individuals, as well as people that use medical devices or have had medical device implants. Candida albicans typically forms biofilms around implanted parts in wounds. These biofilms, which are highly structured sheets of different cells held in an extracellular matrix, are especially resistant to chemical and physical attacks. Further study into this pathogen will help the demographic of individuals who are at risk for hospital-borne infections. Due to its prevalence and ease of use in a laboratory, Candida albicans is a model organism for studying fungal pathogens. As a result, Candida albicans research is done worldwide. A large number of entities study and fund this work. They range from the smaller groups, like Cal Poly Pomona, to large foundations like the National Institute of Health and the National Organization for Rare Diseases. Much of the research that we used had the NIH as a resource. Thank you for watching and good luck on your immunology final.